This presentation is an introduction to organic compounds, specifically carbohydrates and lipids. There are several different ways to classify compounds in the body. A metabolite is any molecule that can be made or broken down in the body. Of those, the ones that have to be taken in through our diet are called nutrients. These can be divided into organic and inorganic compounds. Organic compounds have carbon and hydrogen as the primary elements. An inorganic compound can contain carbon and hydrogen, but they won't be the primary elements. Some examples of organic compounds are carbohydrates, lipids, proteins, and nucleic acids. Some inorganic compounds are salts, acids and bases, and carbon dioxide. Carbon dioxide is CO2. Note that carbon dioxide does have carbon, but no hydrogen, and carbon is not the primary component. This is an introduction into carbohydrates. The building block of all carbohydrates is a compound called a monosaccharide. The three most common monosaccharides are glucose, fructose, and galactose. All of these are isomers, which means that they are compounds that have the same chemical formula, but a different physical structure. Notice that they all have the chemical formula C6H12O6, but their ring structures look different. A disaccharide is formed when two monosaccharides are joined together. One of the monosaccharides is always glucose, and then it will be joined with one of the other monosaccharides. So glucose plus fructose makes sucrose. Glucose plus another glucose equals maltose. And glucose plus galactose equals lactose, which is the milk sugar. Disaccharides are joined by the process of dehydration synthesis and are broken apart by the process of hydrolysis. As you can see, the OH portion of glucose is going to bind with this H from fructose, and then over here that is lost as water. When we break this down, we add the water back in, and that H goes back to fructose, and the OH is on glucose. A polysaccharide is a large molecule with a repeating monosaccharide unit. That monosaccharide is almost always glucose. We can find this in the form of starch and glycogen. Starch is the plant storage form for glucose and is usually found in long, straight chains. Glycogen is the storage form of glucose found in animals, including humans. It is a highly branched compound. Now we turn our attention to lipids. This is what many people refer to as fat. Most lipids are in the hydrophobic category, meaning that they do not react well with water. The building blocks of almost all lipids are fatty acids. These can be saturated or unsaturated, which I will explain in a moment. Almost all fatty acids can be made in the body, but there are a few called essential fatty acids that must be obtained in the diet because they can't be made in the body. A fatty acid is essentially a long chain of carbon atoms bound together. In a saturated fatty acid, each carbon has 4 single covalent bonds, one to each carbon and one on each hydrogen to the side. In other words, it is saturated with hydrogen atoms, or can't accept any more hydrogen. Because of this arrangement, saturated fatty acids are usually solid at room temperature. And see, here's a carbon, there's 1, 2, 3, 4 covalent bonds. So to a carbon, and then to the hydrogens on the side. An unsaturated fatty acid, there has to be at least one double covalent bond between carbon atoms. This makes it unsaturated with hydrogen atoms, or it has the ability to accept at least 2 more hydrogens. When this double bond is present, it puts a bend in the fatty acid. Because of this, unsaturated fatty acids are usually liquid at room temperature. So instead of having 4 single covalent, this carbon has 1, 2 single covalent and then one double bond here. Same thing with this one. It has 1, 2, 3, 4. So there could be another hydrogen here and another hydrogen there. Before talking about triglycerides, let me give you a non-science example of the reason for saturated being solid and unsaturated being liquid at room temperature. Consider that you are packing your belongings. If you had a series of books that were all straight and all roughly the same size, you would be able to pack them very closely together in a box. But if you had several oddly shaped items, they would not be able to fit together nicely, and you would only be able to fit a few of these items in a box. The books, or the items that were able to be packed tightly together, would be a solid, like the saturated fatty acids. The oddly shaped items would then resemble the unsaturated fatty acids. Since you cannot pack them as closely together, they form a liquid. Think about the solid and liquid aspect. What kind of fats are solid at room temperature? And think about what you might have in your kitchen. Which kind of fats are liquid at room temperature? And which ones are usually considered healthier for you, the saturated or unsaturated fatty acids? There are several different types of lipids. The first type we will talk about are triglycerides, or sometimes called neutral fats. A triglyceride is one glycerol molecule and 3 fatty acid molecules put together. When these are being joined, the process of dehydration synthesis is being used, and hydrolysis is used to break apart this molecule. So we have a glycerol molecule and then 3 fatty acids. And notice that we have two saturated fatty acids and 1 unsaturated fatty acid in this example. There are several different forms of lipids that are used as components in the cell membrane. These are usually called structural lipids, and they include steroids, phospholipids, and glycolipids. A steroid is a lipid with a ring structure. Some examples include cholesterol, cortisol, and the hormones such as estrogen and testosterone. Two other structural lipids include phospholipids and glycolipids. They are both found in the cell membrane, but phospholipids are the primary component of the cell membrane. This will be discussed in more detail in a presentation devoted to the cell. They both contain a diglycerol molecule, meaning a glycerol molecule and 2 fatty acid chains attached. In place of the third fatty acid, a phospholipid will have a phosphate and non-lipid group attached, and a glycolipid has a carbohydrate group attached. So here is our glycerol molecule, 2 fatty acid chains. This has the phosphate and a non-lipid group, so this is a phospholipid. Here's the glycerol, 2 fatty acid chains, and a carbohydrate here, so this is a glycolipid. Recall that most lipids are hydrophobic, meaning they do not interact well with water. But the majority of the body is water. So how do lipids arrange themselves to be able to interact with water? They arrange themselves into a structure called a micelle. This is a sphere that has the fatty acid chains towards the center and the non-lipid portion to the outside, where they would react with water. I hope this presentation helps you understand the basics of carbohydrate and lipid structure.